we have been working on genetic disorders. This is where what we call a faulty allele causes disease. And I just wanted to clarify a little bit of terminology here. If someone is affected with the disease, it means they have the disease. An affected individual is someone who has the disorder or has the disease. And we have looked at one set of scenarios. Well, actually, we're going to be looking at several scenarios, but one that we have looked at already is autosomal recessive disorders. And this is where an autosome, remember, this is a numbered chromosome. And an autosomal recessive disorder occurs when the faulty allele is recessive and is on an autosome. So faulty allele, oops, did not mean to put a plural in there. The faulty allele is recessive and on an autosome. Again, an autosome is a numbered chromosome. It's a chromosome that is not a sex chromosome. And we already looked at several examples of this. The examples of this were cystic fibrosis and also sickle cell anemia. And we also saw that in sickle cell anemia, it actually shows some incomplete dominance, meaning that the heterozygous person who is usually has a normal phenotype, normal healthy phenotype, but is a carrier, also has some slight sickling as well. But also being heterozygous also protects against getting malaria, which is advantageous from an evolutionary standpoint to be able to survive malaria. So that's why that recessive allele tends to persist. It is still in existence. But for something like cystic fibrosis, remember we had the dominant allele causes a normal phenotype, but the recessive allele causes fibrosis. The carriers, the heterozygous people, are carriers because they don't have the disorder, but they carry the allele. They are completely normal. So only affected individuals are homozygous recessive. Now, sometimes the faulty allele is not recessive, so that brings us to the next one, which is autosomal recessive disorders. This is when the faulty allele is on an autosome, but I totally messed that up. Fix that. Let's go back and fix this. Holy crap. Totally said the wrong thing there. Homozygous, let's change the color. Homozygous dominant disorders. Homozygous. All right. My mouth and my pen are not working together. Autosomal dominant disorders as opposed to autosomal recessive disorders. We're looking at autosomal dominant disorders where the faulty allele is on an autosome, but it is dominant rather than a recessive allele. So, faulty allele is dominant and on an autosome. Okay, so an example of this. There is one type of dwarfism. And by dwarfism, I mean, I think the medical definition of someone who is a dwarf is someone who is under 5 feet tall, but that's kind of silly. Most forms of dwarfism have result in an individual who is, under like 4.5 feet tall, so very short, very short. Think Lannister from Game of Thrones. Oh gosh, I can't remember the actor's name. Peter, I cannot remember. Sad is it that I remember the character's name, but not the actor's name. But if you watch Game of Thrones, you know who I'm talking about. There are several different types of dwarfism, and I'm not sure what type of dwarfism the guy who plays Lannister has, but there is one type of dwarfism that is caused by a dominant allele. So the dominant allele in this case causes dwarfism, where the recessive allele results in normal height. Peter Dinklage, that's the guy's name. That's the actor's name. Just popped into my head. Who plays Lannister on Game of Thrones. Alrighty. Okay, so of the 3 possible genotypes that we have, someone could be homozygous dominant, heterozygous, or homozygous recessive. This individual will be a dwarf. This individual will also have dwarfism. They will be a dwarf. And this individual here will be normal height. So it's the opposite, pretty much, of autosomal recessive, isn't it? Only here, and here's the deal too. This individual here who's heterozygous, don't call them a carrier. They are not a carrier because they have the disorder. The definition of a carrier is someone who doesn't have the disorder but still carries the allele, still carries the faulty allele. So with autosomal dominant disorders, you don't get a carrier, because if you have the faulty allele, you have the disorder. So let's take a look at this. Let's see. What the heck was the name of that other TV show? Little People, Big World. Let's change the color here. Let's go with this. Little People, Big World, I think it is. Little People, Big World. And isn't it where the parents are both dwarves? They both have dwarfism. But the kids, aren't 2 of the kids normal height and 1 short? So 2 have normal height. 1 is a dwarf. And what's interesting too is that the one who's a dwarf is a twin to one of the ones who is of normal height. And they're not identical twins, obviously. They're fraternal twins. So pretty much just regular siblings. So, knowing this information, what are the genotypes of the parents? What are their genotypes? Let's figure this out. Let's write each parent here. Let's put the mom here, universal symbol for female, universal symbol for male, the dad over here. So these are the parents. And we know that they are both dwarves. So they both have to have at least 1 dominant allele, don't they? So they've each got that one, and we don't know what their second allele is, do we? So we'll put a little question mark there. Some of you have probably got the answer already, and that's cool, but let's look at their kids. One kid is normal height. Kid number 2 is normal height. And kid number 3 has dwarfism. So, what is the genotype of it, the 2 kids who have normal height? And if you said homozygous recessive, ding, 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 you're correct. Okay, the only way to have a kid with normal height is for those kids that individual to have 2 recessive alleles. So they are homozygous recessive, both of them. Well, think about this for a second. Where did they get each of these recessive alleles? I mean, one of them had to come from their birth father and one had to come from their birth mother, so that means that both parents must be homozygous recessive. They had to get their children who have normal height had to get that recessive allele, one from one parent and one from the other, so both parents must be heterozygous for the trait. They must be. All right, now the third child, oops, I'm still an eraser. The third child here, we don't know what they are. They could be homozygous dominant or heterozygous. But let's see what the chances of this are. Let's set up this cross here. Let's cross 2 people who are heterozygous for dwarfism. We've done this before many times. This parent here could donate a dominant allele or a recessive allele. This parent over here could donate a dominant allele, put it at the end of the column, or a recessive allele, put it at the end of the row there. Row, column. And let's fill in the boxes. Just like that. We've done this cross between 2 heterozygous many times. Okay, so there's only a 1 in 4 chance that their kid would have normal height, right? And yet it happened twice. What's the deal with that? Shouldn't they have had, a kid of normal height, and then the next one would have had to have been a dwarf? And then the next one had to be a dwarf? If they have 4 kids, shouldn't only one of them have normal height and the rest would all have dwarfism? And the answer to that is no. It's a 1 in 4 chance every time of having a kid who's homozygous recessive and normal height. Every time you have a kid, all right? So it's kind of like, you know how there's a 50-50 chance of having a male or a female child, right? But it's a 50-50 chance every single time. No one's like, well, okay, our first kid we had a boy, and so our next kid will be a girl. Well, no, you don't know that. It's that second time you have a kid, it's still a 50-50 shot. So it is a 1 in 4 chance of this couple having a kid of normal height every single time they have a kid. And a 3 in 4 chance of their having a kid with dwarfism each time. So we don't really know whether or not their child who has dwarfism is either this box or one of these ones. But they have twice the chances of being heterozygous than they do being homozygous dominant. Here's another example. Well, let's go with purple. We haven't used purple in a while. Another example is what we call extra or a 6th digit. In other words, 6 fingers on your hand. Okay, yes, we know you all. That was terrible. We all have 5 fingers on both hands, don't we? Does anybody here know someone who has 6 fingers on their hand? Occasionally, somebody in class actually does know someone who has 6 fingers on their hands. Yeah, so famously in the movie The Princess Bride. If you've never seen that movie, go Netflix it. It is so awesome. But the 6-fingered man is the bad guy, and they were looking for the 6-fingered man. Well, it turns out that the 6-fingered man had 6 fingers due to a faulty allele. Let's go with the letter F here. The dominant allele causes a person to have 6 fingers on each hand, whereas the recessive allele has 5 fingers. Yeah, for real. This is totally for real. And actually there is, so, okay, you know, homozygous dominant and heterozygous both give people with 6 fingers. And it's only homozygous recessive that causes people to have 5 fingers. And isn't it true that you probably know way, way, way more people who have only 5 fingers on each hand than 6? I'm just going out on a limb here and guessing that. So my question is, or the observation is, if an allele is dominant, shouldn't we see that trait more often? Isn't that kind of weird that we hardly see anybody with 6 fingers? And here's the reason why. It's because the dominant allele is not very prevalent in the gene pool. The dominant allele isn't very common. It doesn't appear very often in the gene pool, in the human gene pool. It's just not a common allele. So my point here is that just because an allele is dominant doesn't mean we're going to see that trait all over the place. It doesn't mean we're going to see more people exhibiting that trait. Sometimes we do, but a lot of the time we don't. Or, you know, we'd all be running around with 6 fingers. I believe it's also a dominant allele in cats. It's a dominant trait in cats as well. You probably know some cats with extra fingers. But there is a population, actually in the U.S., where it's very common for people to have 6 fingers. Anybody ever been to central Pennsylvania, south central Pennsylvania, like on the turnpike from Philadelphia to Pittsburgh? There is something called Amish country in south central Pennsylvania. The Amish, if you're not familiar with the Amish communities, they are a community of people, and it's, I believe it's also faith-based, but they pretty much reject most technology. It's definitely changing, but they try to live as simply as possible. They do not use cars generally. They use horse and buggy, and they tend to wear black with royal blue a lot, and they tend to stay in the communities where they were born. They tend not to move away. They tend not to go, away to college and schools and the like, and they tend to marry within their community. So what happens is that because somebody had this dominant allele, it gets amplified. And so this dominant allele, the dominant trait shows up a lot more because people in Amish communities traditionally, and a little bit, it's changed a little bit over the past 30, 40 years, but they tend to just marry within that community. And so this amplifies the presence of this dominant allele. So it gets amplified. This is actually called, this is a form of genetic isolation. or genetic drift. And it's one of the mechanisms of evolution, so you'll see certain traits that show up in certain populations but not others. So that 6 fingers is more common in the Amish population than out in the general population of the United States. So I don't know, I guess they make a lot of 6-fingered gloves there. Getting fitted for a glove would be a little more challenging than if you have 5 fingers. So that is autosomal dominant. So we have looked at autosomal recessive and autosomal dominant. These disorders occur when the faulty allele is on a numbered chromosome or a non-sex chromosome. But sometimes the faulty allele is on a sex chromosome, and we actually call those X-linked disorders. So this is what our third one that we're going to be looking at, X-linked disorders. So this is when the faulty allele is on the X chromosome. There are a few Y-linked disorders, but for the most part, there are far more X-linked disorders, and I'll tell you why. Way back, a couple of lectures ago, I'm not going to get anybody's head spinning by scrolling back through all that, but I drew a little picture of a cell with little pairs of chromosomes. You know, like we'll have a pair of chromosome 1s, one that you got from your birth father, one that you got from your birth mother. We all have a pair of chromosome 2, a pair of chromosome 3s, a pair of chromosome 4s, stuff like that, on up to the X and the Y. And I drew the X chromosome like this, and I drew the Y chromosome like this. And it's not because I like the X chromosome better. It is because, this is a fact, that the Y chromosome is much smaller. And the X chromosome is much bigger. Way bigger than the Y chromosome. And so the Y chromosome has way fewer genes than the X chromosome. And so just to put out a little question for you, take out this. Can a human live without a Y chromosome? Can a human live without a Y chromosome? Yeah, I sure hope so. I've been doing it all my life. 51% of the population's been living just fine without a Y chromosome. The answer is yes. Females have no Y chromosome. Females are XX, correct? And we do just fine. So you actually don't need a Y chromosome to develop and survive as a human organism. I mean, the genes on the Y chromosome are important for sex determination, but they are not important for survival, whereas those on the X are. So, next question. Can a human live without an X chromosome? Can a human live without an X chromosome? And the answer is nope. Females have 2 X chromosomes. Males have an X and a Y. And I'm talking about sex as I'm not talking about gender. Gender is a socialized concept. Sex determination is physical body, what's going on at the chromosomal levels. So I am talking about physical sex as determined at birth. So female sex as determined at birth have 2 X chromosomes. Males as sex determined at birth, an X and a Y chromosome right there. So females need X chromosomes, males need X chromosomes, everybody needs an X chromosome. You cannot survive without it. And here's why I look up here to the top here where I'm drawing this X chromosome in green up here now. All the genes that are on this X chromosome are absolutely needed in order for a human organism to grow and develop. So everybody's got those, and there's a whole lot of genes on there, and sometimes the alleles for those genes are faulty. So let's just erase all this stuff here. Clean up this mess. There we go. Alrighty, so X-linked disorders are different in that the faulty allele is on the X chromosome. But get this, the inheritance pattern is going to be a little different because females have 2 X chromosomes and 2 chances to get a faulty allele or a healthy allele, and males only have 1 X chromosome where there could be a faulty allele or a healthy allele, and their other chromosome is a Y. So the inheritance pattern is going to be different here. And let's take a look at an example of this with red-green color blindness. There are several different forms of color blindness, but this one in particular is when a person can't see subtle differences in shades between red and green. And if you were in elementary school, in a public elementary school in the United States, you were tested for red-green color blindness, guaranteed. It was where they had you look at these pictures. Like they tell you, like in 2nd grade, hey, we're going to have a test today. You're going to go to the nurse's office and we're going to give you this test. And they give you these things that look like all these bubbles. And what they do is say, oh, can you tell us what you see in these bubbles? And you're like, the number 2, is this supposed to be a hard test? Something like that, and they'll do something like that. You've seen these things. If we were still at HCC, I'd have gone over to one of the labs and I'd have grabbed one of the books to show you. But people with red-green color blindness cannot see subtle differences, not only between red and green, but even between like different shades of those colors as well. Oops, let's erase all this. Gosh, that's fun. Anywho, red-green color blindness is caused by a faulty recessive allele that is on the X chromosome. So the faulty allele is on the X chromosome. I'm just abbreviating chromosome with CHR, and is recessive. There we go. So let's use the letter R for this, for red-green. Why not? But the thing is, we can't say, oh, well, that's the dominant allele and here's the recessive allele. We can't write it that way because not everybody's going to be, and say, oh, someone's heterozygous for this, they're fine. Someone's heterozygous for that, they're going to be colorblind. No, it doesn't really work that way because here's where Mendel's rule about, there are 2 factors. Every individual has 2 alleles for every trait. Some people don't. If there is a trait on the X chromosome, females up here, they'll have 2 alleles for this trait, but males will only have 1 because this allele does not exist on the Y chromosome. So we can't just put R's down there. So what are we going to do? I'm going to change color. Let's go with red. Why not? We're going to put, to show that this allele is on the X chromosome, we're going to draw an X for the X chromosome. And I will let's see. I'm going to put it down here and give myself a little more room. No, do it this way. And what I'll do is put the allele as a superscript on that X. So the dominant allele on the X chromosome is written like this, and this causes normal color vision. So let's just say normal vision. Whereas the recessive allele, the faulty allele, will write as a lowercase r superscript on this capital X showing the X chromosome. This is how we will write the whole allele. And this allele causes red-green, I'll just call it RG, color blindness. There we go. We can't write the Y, well, we can write the Y chromosome, but we can't put an R up there. That's wrong. No, 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 no. Do not do that. Right? The Y chromosome has no allele on it, so we have to leave it blank. This is just a plain old Y chromosome. So every individual is going to have a combination of 2 out of 3 of these, aren't they? So let's look at what our options are. Let's say we could, well, let's make a table. Let's call it genotype in one column, which is what the genes look like, and phenotype in the next column. All right, so let's say somebody could have 2 dominant alleles on 2 X chromosomes like that. So, where'd this go? So is this person male or female? They are female. 2 X chromosomes, born female, in the absence of any other sort of complication. Let's see. So it's a female with what kind of vision? It's a female with normal vision. With normal color vision, that is. I don't know if they're nearsighted or farsighted, anything like that, but they have normal color vision. Okay, or we could have a female with one of each alleles, right? So this is a female. What kind of vision does she have? She has normal vision, but she is a what? A carrier. She's a carrier. All right, do we have any other options for females here? Why, yes, well, yes, we do. We could have a female with 2 recessive alleles, one on each of their X chromosomes. So this is a female with red, green color blindness. Right, so I'm not seeing any other options for females. So let's move on to males. We could have a male with a dominant allele on his X chromosome. And you can't put any R's on that Y. Do not put R's or any alleles on that Y chromosome because it doesn't exist on the Y chromosome. So this is a male with what kind of vision? You got it. With normal color vision. Any other options for males? Sure, we could give this male a recessive allele. So this is a male with what kind of vision? With red, green color blindness. This male would have a red-green color blindness. Yeah. Okay. So who is more likely to be affected by an X-linked disorder? Who? Who? And if you said males, you are correct. Look at it this way. We got 2 options for males, 3 options for females. One out of 3 shots, 1 out of 1, 2, 3 options for females results in red-green color blindness, but 1 in 2 scenarios results in red-green color blindness for males. Think of it that way. Males are more likely to have an X-linked disorder because they only have 1 X chromosome. They only have 1 shot at getting the healthy allele. So if any of you know someone who has red-green color blindness, my guess is that they are probably male. Males are more likely to have an X-linked disorder, and that's because they only have 1 X chromosome. Only 1 chance at a healthy allele. Females have 2, so they got 2 shots. 2 X chromosomes, you're more likely, far more likely, to have a shot. You have more of a shot at having 1 healthy allele than 2 faulty alleles. So, a couple of years ago, oh gosh, it's more than a couple of years ago, probably like 8 years ago, there was someone sitting in this class when we were talking about X-linked disorders and red-green color blindness, and a young woman raised her hand and she said, "Oh, that's my son. My son has red-green color blindness." And we're like, "Oh, interesting." I said, are you sure that it's red-green color blindness and not some other form? She said, no. And we had him diagnosed. He has true red-green color blindness. We're like, "Oh, that's so cool." So we were like, "What is your son's genotype?" And we determined that it was this. Here, right? That is the only way that a male, this is the genotype for a male with red-green color blindness. So here she is. And so her son, let's put him down here. We know that this is his genotype. And she's like, "Yeah, his father has red-green color blindness too." And we're like, "Are you red-green colorblind?" She's like, no. So she has normal color vision. So here's the information up here. Here's the mom over here with normal color vision. Has a kid with has a son with red-green color blindness. And she's sure that it's red-green color blindness. She's like, "Yeah, he got it from his father." And we're like, "Huh?" And I was like, "Class, what do you think?" And they're like, "No, he got it from his mother." And we're like, "Well, okay." So we're like, "Okay, so do you have 2 X chromosomes?" She's like, "Yeah, I have 2 X chromosomes. I'm female," so there we go. All right. And someone in the class said, "Well, are you good with your husband having 1 X chromosome and 1 Y chromosome?" She said, "Yeah." She's like, "But my husband has red-green color blindness." We're like, "Well, okay, what's your husband's genotype?" And we came up with this, with an X chromosome with a recessive allele and the Y chromosome. And remember, there is no R on the Y chromosome. You cannot write an R up here. Do not do that, because it doesn't exist on the Y chromosome. She's like, "My son inherited his red-green color blindness from his father." And we're like, "So how did that work? Did your son inherit his X chromosome from his father or his Y chromosome from his father?" She's like, "Well, duh." He inherited it from his father, right? Because females don't have a Y chromosome to give to any of their kids, right? She's like, but I'm not red-green colorblind. So we're like, okay, so you've got this here, but where did your son inherit his X chromosome? She's like, but then that would mean that I'm... And the whole class started to laugh too, because it just dawned on her that her son's X chromosome came from her ova, right? So she has to have been a carrier, right? Isn't that interesting? Isn't that interesting? So, and it's because we know that males inherit their Y chromosomes from their father, right? Males inherit their Y from their birth father. And males inherit their X chromosomes from their birth mother's birth mom. Period. So, if you know any males with red-green color blindness, they inherited that allele from their mother because it is on the X chromosomes. The X chromosome, males inherit their X chromosome from their birth mother, not their birth father, because if their birth father had donated his X, well then the baby would have been female at birth, right? Not male at birth. So, because that's how the inheritance pattern works with X-linked disorders. Alrighty, so let's take a look at another scenario. All right, so, well, yeah, first up, she's like, this person here was a carrier, and she did not realize it until that moment. So interesting. Okay, so what do we know about women with red-green color blindness? What do we know about females with that red-green color blindness? Here we go. Here's a female with red-green color blindness. Right here. We know that they have to have two copies then. On one copy of the faulty allele on one X chromosome and another faulty allele on the other X chromosome. So let's draw her here. So it is possible for a female to have red-green color blindness. And in fact, my sister-in-law has true red-green color blindness. She cannot see differences there between those two colors. And her husband still has to tell her, honey, that shirt does not go with those pants at all. I was driving around with her one time too, and she's like, oh, these little houses are so pretty, they're pink. We're like, no, they're white houses that you're pointing at and thinking that they're pink. They are not pink. Her color vision is off. What do we know about her parents, my in-laws? What do we know about them? Where did she inherit this one X chromosome, and where did she inherit this one? Well, she had to inherit this one from her birth mother, right? Her mom, or my mother-in-law, right? Okay. And she had to have gotten this one from her birth father, my father-in-law. What do we know about my father-in-law? He had an X chromosome with a faulty allele on it, and then he had a Y, so he was red-green colorblindness. And indeed, yes, I do know that he was red-green colorblind. Now, was my mother-in-law also red-green colorblind? We don't know, but we don't know. We don't know what her other allele is. I'll just put a question mark there because, okay, neither of my in-laws is around anymore. They passed away years ago. And I am sure that my mother-in-law up here would have said that she is not red-green colorblind at all. Although I have to tell you that I saw the color that she painted her bathroom, and I really have to wonder, because it was neon peach. I mean, we're talking neon peach, and she thought it was like this pale peachy beige color. I'm like, oh, wow. It was a scary bathroom. Neon peach. So I gotta wonder, but she would say that she is not red-green colorblind. How do we know if my sister-in-law, if my sister-in-law's mom, my mother-in-law, had red-green colorblindness? What would we look at? Now, somebody right now is thinking, oh, we could look at her parents, but I have no idea. They've been gone for years and years and years and years and years. I can't ask them, and it's probably not in their medical records, even if I could find them. And is there anything else I could look at? And right now somebody is saying my husband, because this is my husband's sister, right? So, husband. There we go. And he's got an X chromosome and a Y chromosome. Where did he inherit this Y chromosome? He inherited it from his father. So where did he inherit this X chromosome? From his mother. Up there. But did he inherit a dominant allele or a recessive allele? For that, we have to look at his phenotype, and I can tell you that he has normal color vision. He is not red-green colorblind, so he has a dominant allele there. He had to have inherited this X chromosome from the same mother of his sister. So, no, my mother-in-law was not red-green colorblind. She was a true heterozygous. She was a carrier of it and did pass it on to her daughter, but passed on a dominant allele onto her son, who is not red-green colorblind. So the neon peach bathroom, I don't know what to tell you. It's might have been a fluke. She was not red-green colorblind, or was she? Or was she? And I'm saying this because even though she's a true carrier right there and shouldn't have red-green color blindness, there is something extra that happens in females that's fascinating. And it is called X inactivation in females. Only in females. It does not happen in males. What happens in females is that during early development of an embryo, in each cell, one of the X chromosomes is inactivated, or it's basically covered up, and none of its genes get to be active. So let me show you how that works. Let's go with red. Let's say this was my mother-in-law's as a fertilized egg, and one of her X chromosomes had a dominant allele and one had a recessive. When this cell divides, it's going to produce two cells with one X chromosome that has a dominant allele and one X chromosome that has a recessive allele. And when these cells divide, you'll have 4 cells, and they'll each have 2 X chromosomes, and one X chromosome will have a dominant allele and one will have a recessive in all of these cells. And then these will divide into 8 cells. You get how this works. 1, 2, 3, 4, 5, 6, 7, 8. And each cell will have 2 X chromosomes, and one will have a dominant allele and one will have a recessive. And I do not know at what cell number stage this happens, but let's say this is the 8-cell stage here. At some point early in development, like let's say around the 8-cell stage, there is something about the cell senses that there are 2 X chromosomes present and that really humans only need one of them, right? Because males have only 1 X chromosome and they do fine. And what the cell does is that it randomly inactivates and covers up one of those X chromosomes. And it's random which one it is in each cell. So I'm just randomly covering up some of these like that. Oop, I didn't put an R there. Okay, well, let's say that this cell here goes on to develop to be the rods and cones in the eyes. The X chromosome with the dominant allele is active, so this person would not have any red-green color blindness. But what if this cell here is the one that goes on to form rods and cones in the eyes that allow you to see color? The recessive allele is active, and this person could possibly be red-green colorblind. It's very interesting. So it's possible that in females you could have some slight red-green color blindness, especially if both of these went on to form rods and cones in the eyes. You could have some red-green color blindness depending on which chromosome is inactive. A really cool example of this is in coat color in cats. So for example, in cats, an orange fur, orange coat color, is caused by an X-linked allele. And cats with a dominant O allele have orange fur. Or will likely have orange fur, it depends if it's male or female. And cats with, I'll just draw a dot for a lowercase o so that we can see the difference, will have another color fur, usually black. So let me go back to black here. All right. So let's say that there's a female cat that has one of each. And when her cells get to the 8-cell stage, I'm only going to draw 4 here, for heaven's sakes. You don't need to be watching me draw X's and O's. Can you see that when we have X inactivation, let's say this one gets covered up, 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 that this cell here will go on to form black fur. But this cell here, because its orange allele is active, will form orange fur. Ever see an orange and black cat? What are they called? They're called tortoiseshell cats, or sometimes they're calico cats. So calico cats are always female, and they're always heterozygous at the O gene right there. So isn't that fascinating? So that's why that's why there are only female calico cats. It's due to X inactivation. All right, so we're just about to wrap up the semester here, and there's one more set of genetics problems that I want to talk about. Let's just get a new color for this. No, let's use green. Because for this, I want to go back to plants. So we'll go back to pea plants. And I want to look at 2 traits at a time. Like we've already done things that are like, okay, if you take a tall tall pea plant that's heterozygous and cross it with another tall pea plant that's heterozygous. You know, yeah, we did all this stuff. It's going to look like this. We've done this like 30 times already. Blah, blah, blah. These will be the short plants, whatever. I want to give you a new trait. So in peas, in pea plants, a dominant allele, capital G, causes the peas to be green. Green plants, we got that. Yes, peas are green, most peas are green. But there is a recessive form of the allele that causes them to be yellow. I have never eaten yellow peas, but apparently they do exist. And it's caused by a recessive allele. And we already talked about how a capital T causes a tall plant and a lowercase t causes there to be a short plant. So I want to do some problems. I want to do a problem where we're working with two traits at the same time. So for example, what if we cross a plant that is homozygous dominant for tall and homozygous dominant for green? Okay, so this is going to be a tall green plant, the jolly green giant of plants, and we're going to cross it with a short yellow plant. So for a plant to be short, it's got to be two lowercase t's, and for it to be yellow, to have yellow peas, it's going to be two lowercase g's. If we cross these, we do not need to do a box to know that this plant over here can only donate a capital T, can only donate a dominant allele, T allele to its babies, and that this short plant here can only donate a recessive T to its babies. All right? And this plant here can only donate a dominant G, and this plant here can only donate a lowercase g. 100% of the babies will be heterozygous at both genes, right? And there'll be 100, oops, what did I do there? Wow, what did I really do there? 100% tall and green. But what happens when we breed two heterozygous plants with each other? What happens when we take two plants that are both heterozygous at both genes and cross them with each other? Well, this plant over here can donate a dominant allele, T allele, or a lowercase t allele, a recessive T allele, or it can donate a dominant G or a recessive G. It could donate a dominant T and a dominant G, or a dominant T and a lowercase g, and oh my God, this one's all the same, does the same thing. It's a mess. We need a Punnett square for this, but we need a way bigger box. We need a 16 square box. 4 square box is not going to do it. So I'm going to scroll up here. Draw me a box. Did not mean to do that. So, you know, you can draw your big 4 square, but then you need to divide up the remaining squares like this so that it's 4 by 4. So 4 across, 4 columns, 4 rows. And remember, we are crossing this right here. All right, now what we need to do, okay, so we need a 16 square box, got that. What we need to do is up here, we're going to put the combination, up at the tops of these columns, we're going to put combinations of T's and G's. Like we'll put a T and a G, a T and a G, a T and a G, and a T and a G that are possible from one parent. And over here, we'll put a T and a G and a T and a G and a T and a G that are possible from the other parent. Whatever you do, do not put 2 of the same letter up here, ever. Do not put 2 T's here, do not put 2 G's here. Do not do that. it will not work. Okay, but how do we figure this out? Let's do this. Okay, so here's the way I do it. I take this one right here. So look over here. And I'll say, okay, this plant can donate this T to its babies along with this dominant G. So I'm going to put a dominant T and a dominant G at the top of this column. Or it could donate this dominant T with a recessive G. So a capital T and a recessive G go at the top of this column here. Now, I don't see any other possible combinations of this dominant T to go with any other G. That's it. That's all we got. So I'll move on to this lowercase t. This plant here could donate a recessive T and a dominant G. So I'll put a lowercase t and a dominant G. Or it could donate a recessive T and a recessive G. And I'll put them right there. Notice each of these has a T and it has a G. It doesn't have two G's, it doesn't have two T's. That's it. So now let's move on to the other parent. And it's going to be the same thing because it's the same as this one, right? But let's just do this again. So this plant could donate this dominant T allele with this dominant G. So, and this time I'll put it at the end of a row, capital T, capital G. Or it could donate this dominant T to its plant babies and a recessive G. So I'll put a dominant T, recessive G. Let's move on to the lowercase t now. So a recessive T could be given to its babies and along with a dominant G. So I'll write that at the end of this row. Or this recessive T and the recessive G could be donated to its plant babies. So now we just need to fill in our boxes here. But here's the deal. Don't put TG, TG. That's wrong. We need to keep the T's together and keep the G's together. So I'm going to write capital T, capital G, capital G, because I take what's at the top of the column and at the end of the row. So take a couple minutes to fill in your box. I hope you're taking notes. All right, and here in class I usually give you a little extra time to fill those out, but here we are. Here it is. You can always stop the video. Let's look at what our options are. We could have tall green plants, yeah. Tall yellow plants, short and green, or short and yellow, right? Those are the options. So let's look at all of our tall and green plants. I'm looking going, well, that's tall and green, that's tall and green, that's tall and green, that is tall and green, tall and green, tall and green, tall and green, tall and green. Oh, that one's tall and short, so no. Tall, short, that's short and green, short and yellow. 1, 2, 3, 4, 5, 6, 7, 8, 9. There are 9 tall and green plants. Tall and yellow. There, that's tall and yellow. That's tall and yellow. Ah, that's tall and yellow, so 3. Short and green. That's short and green. Short and green. Short and green, 3. And the only one that's left, this little one here, yellow. So this is out of 16. So there's a 1 in 16 chance of this cross, which is actually called a dihybrid cross. 1 in 16 chance of this cross producing a short yellow plant. All right. This is actually called a 9:3 to 3:1 ratio. 9:3:1 ratio. And it's for phenotypes. But on the final exam, I might ask you a genotype question. I might ask you, what are the chances that this cross would result in a genotype that is the same as the parents? So we'd be looking for this. So you'd have to go find those through here, somewhere in here, whatever, and find that genotype. So please make sure you pay attention to whether or not I'm asking you for phenotype or genotype on the final exam. Okay, so speaking of the final exam, this is the final lecture. I'm going to give you next week time to catch up, because I noticed that not a lot of you have even watched the last lecture, and I'm just cutting you all a break by not throwing even more information at you. So I'm going to give you some time to study and get all of your notes and rewatch lectures as you need next week. And so we will have our final exam coming up. Oh, let's just use hot paint just for hoots and grins. Okay, so finals week is May 18th through the 21st. We would normally have our final exam on Tuesday the 19th. So I'm going to give you 2 days. Final exam. It will open on May 19th at, 100 hours, a.m., whatever. That's midnight, but if I say midnight, somebody's going to think that it's, the night before or something like that. No. So at 100 hours on May 19th, that's a Tuesday, through May 20th at 29, what the heck? How am I giving ourselves extra days and extra hours in the day? At 23:59, 100 hours p.m. That is 1 minute till midnight. So you have all day on the 19th and all day on the 20th to take the exam on Moodle. You will have 75 minutes for it, unless you have an IEP and you let me know. IEP get double time. It will be about the same length as your previous exams. So, 35 questions or so. There will be a lot of multiple choice, short answer, an essay or two. You will have a lot of genetics problems on this too. You'll have some essays. And I can't believe I have to say this, but based on the ridiculous amount of and unacceptable plagiarism on that last exam. Okay, last exam, I found you plagiarized, and there were some other instances of it too that I barely caught. You cannot copy and paste from this video, from anywhere on the internet, and get credit. Okay, so, and last time you got a 0 on questions where I found you plagiarizing. You will get a 0 on the entire exam if you are found to cheat. And that includes plagiarism. And by the way, plagiarism, I am not required to tell you what plagiarism is. You all know what plagiarism is, and you all know that it is in the student handbook. It is presenting work that is not your own. If you copy and paste something and post that in your essay, you're getting a 0 on the exam. That is plagiarism. You're getting a 0 on the entire final exam. If you change like 1 or 2 words, That's still plagiarism. You cannot just change the word crucial or leave that out and say NAD is a crucial molecule, blah, 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 and have the rest of the sentence be literally copied and pasted. That is still plagiarism. That is using words that are not your own. So left a hole in my heart that only bacon can fill, seeing you all doing that, and I will not hesitate to give zeros on the entire exam if you are found to have plagiarized on that. So yeah, that nice little aside. Let's just change color for a little bit here and talk about, now that we know when the exam is, let's talk about what will be on it. About 60% will be new stuff. So since the last exam, so this includes photosynthesis, and it includes all the genetics stuff that we did. Stuff, that makes it sound so cheap, doesn't it? But all the genetic stuff that we have talked about here and done problems for. So, you know, your basic Mendelian genetics with dominant and recessive stuff, and complete versus incomplete dominance. You'll have 1 trait problems and 2 trait problems like we just did. You will see autosomal dominant questions, autosomal recessive questions. You will see X-linked questions, all of this stuff. Codominance, multigenic traits, you need to know what those are as well. So all the stuff that we have been covering, you will definitely have problems for each of these types of things. So human genetic disorders right there. Oh, X inactivation, epistasis as well. So all that stuff, and about 40% old. So everything from the beginning of the semester. I might ask questions from previous exams. I might not, just because we are online now, but you can review your first exam that was handed back to you already before the pandemic, but this will include everything from, you know, protons, neutrons, and electrons, and all that up, all through cellular respiration. So please review your notes and make sure you can find everything in your notes. This is an open note test, so you are able to look things up in your notes, but you just can't copy things from the internet to answer these questions there. So yeah, it'll be everything from the semester forward, but it's only going to be about 40% of the old exam. Just a heads up, there will be one more thing. I'm giving you, it's not an assignment, but I will assign you a video to watch, and I will put that up on Moodle. Okay, so a video on what's called epigenetics. I will put this on Moodle for you. It's not my video, and I didn't want to just link it here. I wanted to make sure that it was linked on Moodle as well. There is like a 15-minute video that I just want you to watch. I'm not going to test you on the teeny details of it, but you need to have a general idea of what epigenetics is. So it's a fairly basic video, and I will ask you a question just to make sure you watched it. So please make sure you watch that video that I link on Moodle. And I will be available to answer some questions that you have next Tuesday. Since I'm not putting up a formal lecture here, I want you to watch this epigenetics video instead, and I will be available for questions on the 12th. I hope you're having a great week. I hope you're staying safe. I hope you're enjoying some really nice weather wherever you are. Take it easy. It's been a pleasure. It's been a crazy semester, but it's still been great to be a part of your educational and biological journey. Take it easy.